Good afternoon. Some of you may realize I'm not Stan Nelson. Stan got called away for something urgent. So it's take this opportunity to introduce a longtime colleague and friend. You know, in science, you interact with some people over and over again over a period of years, and I've had the fortunate the good fortune to interact with Joe on several of these talks and visits. Joe is the Rady Professor of Human Genetics and Neuroscience, or something like that, at UCSD. That's one of our satellite campuses down south. And Joe, so Joe is a Valley boy, although ChatGPT says you were born and raised in Ireland.
Oh.
And it's which was fun, and went to UCSD undergrad, then went to medical school at the University of Chicago, and then moved to Boston to do his pediatric neurology training at Boston Children's in the days of the Giants, and he and then did research training with Chris Walsh and made really breakthrough discoveries in the mechanisms of aberrant neural development, discovering the double cortin, the cause of some neuronal migration disorders. He took been faculty at UCSD for a number of years after a brief stint at Harvard, and has really, we use the word pioneer a little too frequently, but Joe is a pioneer in the study of the genetic basis of neurologic disorders, ranging from abnormalities in brainstem development to really seminal work that hits very close to home here at UCLA, discovering the molecular underpinnings of cortical dysplasias, of focal cortical dysplasias that cause epilepsy, and that's had a dramatic impact on how we think about these patients, and certainly touches on some of the work that I'm interested. Joe has been enormously productive, has a large number of awards that I was too jealous to go over. If you think about productivity, if you search him on PubMed on my computer, he had 34 pages of results, whereas if I search Kornblum H, it was about half of that. And some of that is there's some sociologist named H. Kornblum that contributed a few of those papers. But Joe's, consistently been a leader in the field, and it's great fun to have him here. And I think we're in for a treat. So thanks for coming out.
Thank you. Okay. Thank you, Harley. It's so nice to be at the mother campus. No, UCLA is wonderful. I grew up in the Valley and used to spend weekends here in Westwood. This is where all the action is. So it's really great to visit. And I'm really pleased to be participating in this seminar series for Precision Health. I think a lot about precision health, and I really admire what's happening here at UCLA. So we'll be talking about pediatric brain diseases, which is my focus, and there'll be kind of two stories. And I just want to give you a little bit of background about what we're doing in some of our recent studies. So I have some disclosures here. And our research focus, we have four main areas. One is in recruitment of special populations of pediatric brain disease patients. We study mosaicism and use that then to understand human development. We're interested in the molecular mechanisms of spina bifida, which is the most common CNS structural condition. And we're using stem cell models to develop novel therapies. So I thought I would start here at UCLA, where we have had a long collaboration with Gary Mathern, who was head of pediatric neurosurgery here. And I remember in my training hearing about this disorder called hemimegalencephaly, where half of the brain is enlarged. And it's a disease that never really made any sense to anybody, because how can you have half of your brain enlarged? So this is one of Gary's patients. These are identical twins. And this is the MRI from the healthy twin. And this child has severe epilepsy. And this is the healthy hemisphere. And there's one of the hemispheres here on the left side is massively enlarged. And what could produce this? We didn't have any good idea, but the treatment for this condition is to resect the abnormal hemisphere. And Gary Mathern became kind of the world's expert in hemispherectomies. And this would cure the child of seizures. And so he had a whole freezer full of hemispheres he had resected from children with this condition. So I was interested in this and thinking, well, this could be a genetic condition. The only thing that made sense is maybe there was some mutation that developed early in development that gave rise to this abnormal hemisphere. And Gary was very happy to collaborate. And we found the causes for this condition, and it led to discoveries of some of the major causes for focal brain dysplasias. This accounts for probably a third of all pediatric intractable epilepsy, where on the brain MRI scan, there's a patch of cortex that didn't develop properly. It's dysplastic. And now the treatment for an ever greater percentage of these patients is to resect the brain. We, over 10 years or so, built a cohort of 327 patients on whom focal resections were performed, a fraction of which came from UCLA, but all over the world. And this is a paper that we published last year. And these are the most commonly mutated genes. There's 8 genes that are recurrently mutated. These mutations are in a small subset of cells in the resection, sometimes just 1 or 2%. So we had to really develop better methods to find them. And what you can appreciate is on this uncle plot, there's a lot of genes that are listed here that are seen just in one patient. So these are mosaic mutations, genes carrying mosaic mutations. We think they're the cause, but our cohort has to grow still larger so we can determine if these genes are recurrently mutated, seen in more than one patient. So we'll have confidence that they really are the cause. But any of these mutations that we put into mouse and drive into developing cortex, we can see focal brain dysplasia. So we think that they really are drivers of these focal brain dysplasias, but I think there's just so many genetic causes for focal brain dysplasias. And this is kind of a theme of the talk. So there's certain of these genes, if they were mutated in every cell in the body, probably it would not be compatible with life. But humans can tolerate mutations in a small percent of genes. They can lead to tissue dysplasias, they can lead to cancer, obviously. And I think that focal cortical dysplasias is similar to cancer in that regard. So we have genes that are really essential that when mutated in a small number of cells give rise to focal brain dysplasias, and then we can find them after resection. This has also given rise to some new therapies for focal brain dysplasias. What we think is happening is that the mutation occurs after fertilization. So you have the father and the mother's germ cells, they meet to make the zygote, and then that zygote, the cells divide, and then there's a mutation that occurs in one cell, and that cell then can populate the abnormal part of the brain. And in this way, these are considered somatic mosaic mutations. We got pretty good at identifying these mosaic mutations as the technology developed over the last 5 or 10 years or so. And we then used that ability to identify mosaic mutations in healthy individuals. Because every time a cell divides, there's at least one new mutation that occurs. So we can use those as cellular barcodes and answer questions about human development that really we could not answer previously. Of course, we take a lot of lessons from mouse models. They're great for a lot of things, but humans have, where the rules might be different. And so in this paper that we published 2 years ago, we use these mosaic variants that appear in healthy individuals to reconstruct how the human brain was put together. And what we found was that the human brain is assembled with over time, there are restrictions in how the cells can move around the brain. The first is a cleavage between the two hemispheres. This happens very early, and this would not have been predicted from the mouse work. So for instance, we can see these red cells here, there's mosaic variants that are present in these red cells on both sides of the brain. And then you might develop a new mutation in this blue cell here. This blue cell can't spread to the other side because this occurs after the cleavage of the two hemispheres, this restriction of cell movement. And then later there's an anterior-posterior separation that happens. And so the orange cells get stuck up here just in the, one lobe. And we think this is just giving us important clues as to how the human brain is assembled. And we do this work from healthy human cadavers that we access through our university pathology service. And as we've gotten better at these methods, we can then tune our questions more precisely to look at things that maybe were supposed from animal work, but maybe have never been tested before. There were a couple recent papers that suggested that the interneurons in the brain, these are the neurons that produce the GABA, that set the inhibitory tone in the brain. In mouse, they're all derived from subcortical regions. So this is a mouse cortex where all the gray matter is located. And all of the interneurons here in red derive from the ganglionic eminence and migrate up into the cortex. There was some evidence in humans that perhaps, or at least in non-human primates, that some of the inhibitory cells could come from the same progenitors that give rise to the excitatory cells. So in mouse, the excitatory cells are here in green, and they derive from progenitors that line the ventricle. And using these mosaic variant barcodes, we determined there are two sources of inhibitory neurons in the human cortex, one that comes from the subcortical region and migrates up, just like in mouse, and another that derives from the same radioglial progenitors that give rise to the excitatory cells. And it seems it's about half of the inhibitory neurons in humans come from a dorsal source, and this was published this year. So we're interested in asking other questions using these mosaic variants in healthy humans. But I wanted to turn our attention to a disease that we recently started a study called spina bifida. Meningomyelocele is the other name for it. It's the most common CNS structural disorder. And I got interested in this because I was looking around for a new project and realized there's almost nothing known about this. It's very common. It's an ancient disease. Two of the mummified children found in the tomb of King Tut had thought to have spina bifida. You can see the splayed vertebrae here in the X-ray of one of them. And so we thought we could study this and try to make inroads into the causes, and maybe we could develop better therapies for preventing the disease. So spina bifida, as I mentioned, very common, 3 in 10,000 live births. There's an open spine, exposed neural tissue. And oftentimes this is picked up on prenatal ultrasound. And we see the rates increasing actually over time now as there's some limitations in accessing abortion services across the US. But a lot of these babies are carried to term, and they're delivered generally by C-section. And there's a sac that contains the herniated spinal cord. And there's additionally hydrocephalus seen in most of the cases. That's water on the brain. And we know that folic acid can reduce the risk by about half. We don't really understand the mechanisms. So we thought this was a great project. No one had really studied this in any serious way to understand what could be driving it. Well, we knew there was a continuum of this. And this is the most severe form here that's compatible with long-term survival, but there's milder forms called meningocele where the spinal cord is intact, but there's a sac of fluid. And then there's the mildest form, which is called spina bifida occulta, where there's bony abnormalities, like this is missing the posterior process. And this is present in 10% of all humans, mostly asymptomatic. What we don't yet know is whether these are really part of the same continuum or really they're separate conditions. So we'd like to get at that over time. As I mentioned, spina bifida has hydrocephalus frequently seen, and it's associated with something called Chiari-2 malformation. You might know there's Chiari-1 and Chiari-2. Chiari-1 is generally seen in healthy individuals that presents with a herniated cerebellar vermis into the foramen magnum, and this is associated with headaches or focal neurological findings. Chiari-2 is seen with spina bifida. You can see this is the open neural tube here, and this is the hydrocephalus that's present, and there's a herniation of the posterior fossa contents into the foramen magnum. But it's very different from type 1. The neural tube starts off as a neural plate. You probably remember this from embryology. And we can't see that in mammals because it's happening in the womb, but we can watch in species like chick, actually watch the neural tube close. And this is a short movie that was made by a collaborator from Australia of the chick neural tube closing. And you can see it kind of zips up. It's really a beautiful phenomenon to watch. And this is very complex. You know, there's cells moving around, they're touching one another. This is not going to be a single gene disorder. We know it's way too common to be a single gene disorder. So we wanted to understand the basic mechanisms. So we have the neural plate that happens early in embryogenesis. These cells then loop up and connect dorsally and close the neural tube. And this fails to happen in patients with neural tube defects. Heritability is somewhere in 70–75%, so it should be genetic. There's after the first baby is born with this, there's 100 times increased recurrence risk, but it's not Mendelian. And there's about 200,000 patients in the US with this, and there's many mouse models that have neural tube defects, and they include folate responsive neural tube defects, but the genes that are mutated in mice haven't panned out in humans. So there seems to be different kind of causes in mouse. So we had a very specific hypothesis. We wanted to test whether there were new mutations. I was really struck by the work in the autism field where by studying trios, so father, mother, child, where the child has a disease, a condition, by subtracting away the variants in the parents, we could find just the new mutations that have occurred in the baby, and that's called de novo mutation approach. And this makes a very important contribution to pediatric disease. And the reason is because many of these children won't pass the mutation on, so they're under strong purifying selection. So the only way to really find them is looking in trios like this. We can't use typical pedigrees to look for them like the field of human genetics was built upon. And so we put together a spina bifida sequencing consortium in collaboration with the Spina Bifida Association, and we recruited around the world from different parts of the world where they have sometimes higher rates in the US, sometimes lower rates. And we used a trio approach, and then we did genome sequencing of the father, mother, and the child, and we looked at both de novo and single nucleotides and structural variants, and then did statistical analysis. And we used a control cohort, the Simon simplex controls of 732 to benchmark against, a total of 4,614 exomes, and now more genomes, and then looked at mutations. And this summarizes the result. This has not yet been published, but it's in preprint. We found that there was a dramatic enrichment for likely gene-disrupting mutations in patients here that are shaded compared with controls. So it's more than double the rate seen in patients. This was the case for gene-disrupting mutations. It was the case for de novo missense mutations in what are called HC genes, highly constrained genes, whereas the synonymous mutations were equal in the two cohorts. So just to orient you, the left side of this are the patients and the right side of this are the controls. So we can see there's a shift away from the middle in patients. And overall, we can conclude that around 25 to 50% of what looks like a damaging mutation in the meningomyelocele cohort are mutations that contribute to risk. And with a cohort of around 800 like we had, we thought we should be able to identify 10 to 20 recurrently mutated genes. And this will be a big breakthrough because there's almost no recurrently mutated genes in spina bifida except for a few recessive causes that have been identified. And this summarizes the data in two ways. So we're looking at all the genes and then just looking at constrained genes that are under strong purifying selection. And what we found is that both groups of genes show a strong enrichment for de novo mutations in cases over controls. And the best example here is with an odds ratio of 5/5. So if you're a patient, you're 5 times more likely to have a de novo missense mutation than you are a control. It means that not every de novo missense mutation that we find in a patient will be contributing to risk because they're found in healthy individuals as well. But if we have enough patients, we can collapse them and find the genes that are mutated and then try to build protein interaction networks. And just as we predicted, we found several genes that are recurrently mutated in this cohort. These are the genes that we found mutated in two or three families, but it's a small percent of the total genes that we found. We found 86 total genes, of which only 8 were recurrently mutated. So there's a lot of single hit genes. But when we look at the genes that were mutated in patients with MM versus controls, we see that they're much more highly connected if they're mutated in a patient. This is the number of edges between the proteins that we found with the mutations compared with controls. And the protein interaction network defines several modules. So we see planar cell polarity, Wnt signaling, neuronal connections, dendrites, protein localization, cell junctions, and something called neural tube closure. This mostly came out of prior mouse knockout work. And I wanted to give you a couple of examples. So this was, we found 2 patients with de novo mutations in a gene called PAX3. This is the mutations that are both stop codons. And we found one of our patients had an inherited mutation. That's listed here, the last one. So PAX3, you might know, is mutated in a condition called Waardenburg syndrome. It's a paired Bach transcription factor. And this is the location of the mutations that we found. And I should mention that in OMIM, if we look in the online Mendelian Inheritance of Man database, there's 4 cases of Waardenburg syndrome that have spina bifida. But, you know, the connection's pretty weak. And this is Waardenburg syndrome. It's one of the most common causes of sensorineural hearing deafness, 3% of all cases. And there's also these pigmentary abnormalities with this white forelock and either heterochromia, where the irises are 2 different colors, or you have what's called brilliant blue irises, like seen here in these 2 kids. And also Hirschsprung's disease, cleft lip and palate, and occasional meningomyelocele, like I mentioned. So this was the child that we recruited from San Diego. She has spina bifida. She's in a wheelchair. And this was her underlying diagnosis. And when we found the PAX3 mutation in her, it's a stop codon, we went back and we said, well, she should have Waardenburg syndrome because this is fully penetrant when you have a haploinsufficiency for PAX3. But the parents said she'd never been diagnosed with Waardenburg syndrome, but she is deaf. So that was a clue. I think somebody had missed Waardenburg syndrome because she has hearing aids, you can see here. And we can see she has these very bright irises. So I think this is what we're likely to find with our cohorts. They had missed Waardenburg syndrome. She had the PAX3 mutation that we found, so we can say she actually probably has both. But in this case, the spina bifida really stood out as the predominant feature. So that was her underlying diagnosis. But as I mentioned, in this family, the father was a carrier for the mutation, and he was healthy. He had no reported symptoms, and this was him. So we wondered, he didn't have any symptoms. He didn't have hearing aids. He was healthy. He had no spina bifida. His irises seemed to be normal color, and he was bald, so we couldn't see if he had the white forelock. You know, this was the problem. So we went back to he gave us his mother's number, so we got a picture of him when he was a teenager. And he actually has the white forelock. So we think he's a kind of a form fruit of Waardenburg syndrome. And this is human genetics. This is really, you know, we're dealing with partial penetrance and reduced expressivity. And we see just the tip of the iceberg. But we have to let nature guide us in how we make discoveries and how we put these, understand the genetic architecture of these conditions. Another thing that we found, we were quite surprised, was 1 or 2% of our cohort had de novo mutations at chromosome 22q11.2. This is chromosome 22, and this is actually the most common structural variant that's clinically relevant in humans. It's present in maybe 1 in 3,000 live births. And it's a very common cause for, of course, DiGeorge syndrome, velocardiofacial syndrome, but also one of the most common causes for schizophrenia and other psychiatric conditions. So we found here you can see something like 8 or 10 patients with de novo mutations in this locus, which is much higher than we'd expect by chance. Out of 800 to find 8, that's 1%. It should be present in 1 in 3,000. So the odds ratio of finding this was around 20-fold more common than expected by chance. And these are the sizes of deletion. So this region here, there's 4 low complexity regions called LCR, A, B, C, and D. And the classic seen in DiGeorge syndrome is the 2.5 megabase. We found a couple of families that had the 2.5 megabase, but we found a couple of families where there was a much smaller deletion down here between C and D, which is not part of the critical region for DiGeorge syndrome. And there's 10 genes in the interval here. So I should say these families, we went back and rephenotyped them, and they look like they do have DiGeorge syndrome that was just missed. they were diagnosed with spina bifida. But these down here with these smaller deletions had pretty normal face, no evidence of cardiac abnormalities. And so we think that there's one of these genes down here is increasing the risk for spina bifida, but we didn't know which one. This was a paper that was published a couple of years before we encountered. This was a family where the mother carried a deletion of chromosome 22, and then she had 2 children with the atypical deletion that I described that had spina bifida. And so with this, we figured that maybe this deletion is really important in spina bifida risk. And so we decided to set up a collaboration with the CHOP, that's Children's Hospital of Philadelphia. They have a center of excellence for chromosome 22. They have like maybe 2,000 patients that they've recruited over time. And they were enrolled based upon the presence of the deletion. So we asked them if any of them had spina bifida. And they said, well, no one's ever asked us that before, but we'll go back and check. And they found many more patients than you'd expect by chance in their cohort. So what is this, like 8 or 9 patients among 2,000? So that's also an odds ratio of about 15. So if you have the deletion, you're 15 times more likely to have spina bifida than expected by chance. If you have spina bifida, you're 20 times more likely to have the chromosome 22 deletion than expected by chance. These are very strong correlations. And so we set about to try to find the gene in this interval that was mediating this effect, and we started knocking out these genes one by one. And we started with these genes, and we didn't find any spina bifida in any of them. And then we were contacted by Dolores Lamb at Cornell, and she said, I'm working on the crackle knockout mouse, and we see there's low penetrant spina bifida. So we said, aha, that must be the gene. And that's the gene here. We hadn't got to it yet, but she really helped point us in the right direction. And so we started working on the crackle gene. So crackle is an adapter protein. It's kind of a modular scaffolding protein. And Dolores had found that there's a low incidence of what curly tail, which is a kind of a mild phenotype in mouse embryos for neural tube defects. And she found there's some spina bifida. This is an open neural tube in a mouse in the knockouts. And a low percent of them had exencephaly, which is an even more severe form of neural tube defect you can see. But these were low penetrant. So among 52 plus 15 minus mice, we had less than 10% of them had any neural tube defect. And this was on a normal diet. So we said, well, maybe we know that folic acid plays a really important role in this disease because we know that folic acid reduces the rate by about half. So we put those knockout mice on a low folic acid diet, and 2 things happened. First, the penetrance went up dramatically. So now almost 40% of the mice had neural tube defects. And second, the milder phenotypes of curly tail went away. So we think the effect is to push the severity and the penetrance as on a low folic acid diet. And we think this is giving us some clues as to how folic acid is preventing disease in humans. But we don't think of crackle really as a neural tube defect gene. It's a modular scaffolding protein. And only like I said, half or even less than that of the knockout mice had any neural tube defects. We figured there should be something present in all the knockout mice. So we went back and very carefully analyzed the neural tube of these crackle minus mice. And every single one that we looked at, we found some evidence of defective signaling that known to be where crackle plays a role, such as ERK activation using phospho-ERK antibodies. We can see in normal mice, a phospho-ERK is along the lining of the ventricle of the spinal neural tube, and in the knockout mice, we see it's very abnormal. It's also involved in ABL expression. So we stained ABL, and we saw in the heterozygous mice, ABL is expressed right here along the lining, and in the knockout mice, we see it was abnormal. So we think that, and this is also another clue as to how spina bifida. These are not spina bifida genes. These are genes that are involved in basic mechanisms of signaling. And there's a small percent of people who have these mutations that develop spina bifida. And so this gives us kind of a model of thinking about threshold of disease, where we have some mutations like 6, a gene or a mutation that might be so strong it's going to produce spina bifida no matter the environment, whether it's folic acid enriched or folic acid depleted. But there's other mutations like number 5 here that might be in a different gene or a different allele. It's not severe enough to push you over the threshold for disease unless you also have folic acid deficiency. And that's here indicated by this risk bar in green. And together, the gene and environment are interacting to determine the risk. And this is, we think, the future of studying these cohorts. So to conclude this part of the talk, spina bifida is a very complex disease. We see an enrichment of these likely gene-disrupting mutations. Folic acid may protect humans from the effect of mild mutations, and the genes identified so far suggest altered cell biological processes. And we still don't have a lot of the answers, like what are the cell types that are involved here? Can we predict future spina bifida genes? We like to develop some of these questions further. So maybe I'll just stop here and see if there's any questions on this part, because the second part of the talk is kind of unrelated to this. And I didn't want to distract in case there's any questions. Yeah? That's what we're trying to look at exactly. Are there other mutations in trans that could set the risk? We're looking at that exactly. So we have now a cohort of 22q deletion patients with spina bifida and without spina bifida. And so we're looking at mutation burdens from genome or exome sequencing to hopefully we'll see something. We haven't so far, but we know that inherited mutations will play some role, but so far we haven't uncovered any of them. Yes. Say again? Yeah. Oh, the mTOR, yeah. Oh, mTOR, from Gary Mathern's collection, we found one patient with an mTOR mutation. Most other patients had mutations in genes in the mTOR pathway, but it turns out we were undersampling for mTOR. And now, in retrospect, we can see mTOR is the single most commonly mutated gene for focal cortical dysplasias. It's present in maybe 20–30% of patients. These are activating mutations. They're the same mutations that are seen in solid malignancies. And this has given rise to some idea that maybe mTOR inhibitors can be used to treat the epilepsy. And I think that the verdict is still out, but so far the results have not been very convincing. We're not exactly sure why, but mTOR remains the single most commonly mutated gene for focal brain dysplasias. And this is a result of that work. And I should say that that work is then followed up by several other labs internationally. Yeah, question in the back?
Related to some of those variants where they have lower penetrance and they do not really show up as individual counts. Yeah, I'm wondering if the recurrence rate is related to lower penetrance variants like in PAX3 or other genes that may have not been recognized.
Yeah. Yeah, I think you nailed it right there, exactly. So the question is, what's setting the penetrance? We have no idea at this point. The genes that we see mutated in our cohort, we also see they're present with loss of function mutations in the NOMAD database. So in large measure. So we don't yet know if the mutations are special alleles that are maybe gain of function as opposed to loss of function. This is one possibility, and we're trying to model those now in cell culture or in mice. The other possibility is that they're just partially penetrant. And so I think over time, just like with autism and congenital heart disease and cleft palate and these other conditions that have a very similar genetic architecture, we're learning all of those things can happen. And it's exciting times because now we have the power to really test those hypotheses. Thank you. Okay, yes. Yeah. Crackle heter, so this what was the heterozygous mice were that we consider them to be normal because they never have spina bifida. And you probably have picked up that in the humans, they're haploinsufficient. So the zygosity was not matched. But we find exactly the same in mice, heterozygous versus wild type, in terms of staining for phospho, ERK, and things like this. But the knockouts always show these phenotypes. And so we think this is what Crackle is doing, and that's increasing risk for neural tube defects. Okay, so I'm going to get onto the second part of the talk. this is, I think, also some interesting work. We're working on precision medicine in San Diego and thinking about treating patients kind of on the micro scale. And I was really impacted by this paper from Tim Yu at Boston Children's. This was a girl named Mila, and maybe you know this story. So she had a very rare mutation that gave rise to Batten's disease, which is severe epilepsy. It's lethal. And it's a recessive disease, but one of her alleles was a splice gain. So normally you splice from a GT to an AG, and she had a mutation that gave rise to a novel splice acceptor site upstream of the normal exon. And Tim wisely recognized that this could be approached with an antisense oligonucleotide, which could cloak this cryptic splice acceptor and restore splicing. And this paper he published in the New England Journal proved just that this was an ASO that was targeted for just one patient, because she's the only one that has this mutation that was known at the time. And it really changed the way people in the rare disease field are thinking, wow, what if we could develop drugs that were as precise as this to target the cause, but the drugs might have to be used on the micro scale. So it's really hard to develop small molecules that can work on the micro scale like this. But antisense oligonucleotides are really well suited for this because they're kind of platform technology. You just type the sequence into your computer and they'll deliver you the ASO tomorrow, and you can do experiments really quickly. So they're really nice. And so we started thinking whether we could scale up, some of Tim's really important work that he showed this is possible. And so I'm not sure how much you know about ASOs, but they're just small pieces of DNA, and they bind to the RNA, and they produce effects on gene expression. And there's three main ways that they can work. One is through a splice gain, like I just showed you with that patient, Mila. You can cloak a splice site to correct splicing. We can use them to target RNA to destroy, we can use them to correct splicing if there's a poison exon. These are exons that have a stop codon in them, and they naturally downregulate gene expression. We can force splicing away from a poison exon back to a healthy exon. Or we can destroy the RNA if there's a bad mutation, like a toxic gain-of-function mutation. And here the antisense can be anywhere in the transcript, and it will recruit RNase H to destroy the RNA. And this can be done in an allele-specific way, so we can target just one copy of the gene and leave the other copy intact. And so we started thinking about this technology as platform. And we were also really impressed with this paper that was published in 2020 from the University of Michigan group. Dravet syndrome is a severe form of epilepsy. It's due to haploinsufficiency of the SCN1A gene. And SCN1A is a sodium channel, and it contains a poison exon that traps about half the splicing. And what they showed is that in the Dravet syndrome mouse model, like maybe only a third of them survive to 90 days post-conception. And they can treat the mice with one dose of an intracerebral ventricular antisense that prevents splicing to the poison exon and forces it to the productive exon, and that completely rescues the lethality. Wow. I mean, you don't see preclinical data like this. 2/3 die versus 100% survive. This was really powerful, and this is now entered clinical trials for Dravet syndrome. But it really told us that the approach to therapy really depends more upon the type of mutation, not the type of disease or even the gene. If we can just think about classifying mutations into those that can be targeted with antisense, and we can learn a lot from this, even though it's going to be done at the micro scale. So loss of function mutations, the therapy has to reactivate the gene. Gain of function mutations, the therapy needs to inactivate the gene. Very simple-minded people like me, I like things like this. This is just great. So about the same, now I had this idea, but I was not the only one. Lots of people were thinking, wow. And I was really fortunate to connect with Stan Crook. He was the founder of Ionis Pharmaceuticals. They invented antisense technology there in Carlsbad. And when he retired, he had the same idea, and he started a foundation called NLORM. And the mission is to apply the efficiency, versatility, and specificity of antisense technology to charitably provide experimental antisense drugs to treat patients with ultra rare diseases where there's less than 10 patients that can benefit. And the reason we have kind of less than 10 is we know these will never be attractive to companies. These are not going to ever be kind of for profit. So we just make the antisense and give them to the doctors for free. And I called him up. I said, I love what you're doing. Can I help you? And he said, can you be the chief medical officer? I said, sure. Okay, I'll do that. It's a nonprofit, so I can keep my day job. But I'm learning a lot about antisense technology and the power there. And at Rady Children's Hospital, we have a lot of sequencers. So we really rolled out precision medicine using genomics as the primary driver. So we're doing genomes on lots and lots of sick babies. And now we're thinking about how to treat them. And one way I mentioned is to upregulate genes through antisense. We can treat both loss of function, haploinsufficient, and toxic gain of function mutations. We just have to design the ASO, tweak it a little bit, and then develop it, and hopefully we can treat a patient with it and see if they get better. I want to give you a couple of examples of how these are used. And so now, as my involvement in NLORM, we get nominations from doctors around the globe, actually. We're focused on US patients. And I'm going to show you a couple of the nominations that we've dealt with in the last couple of months and show you how we think about things. So this is a child that has a POLR3A mutation, and this is the OMIM entry. It's a hypomyelinating leukodystrophy, and it's autosomal recessive. And you can see the brain scan here. There's white matter abnormalities and ventriculomegaly. And it's recessive. And so normally recessives can't be treated with antisense because usually it's biallelic loss of function. And we got the doctor sent us the clinical genetics report from the patient. And this is the report here. There was a likely pathogenic, you can see the amino acid substitution from the father, and then a VUS from the mother that was a synonymous change. In other words, it didn't change an amino acid. It was a T to T. So they call that T equals sign. So we got the report and we looked it up in the genome browser. And this is the gene, and this is the actual mutation here. So this is a splice junction site, and we're looking from right to left. So this is unfortunately inverted from how you normally think of splicing, but bear with me. So the normal splice acceptor in this orientation is a CT. Her mutation is a C to T, according to her report here. And so that changes the CC to a CT that looks just like the canonical splice acceptor. So we think what's happening is this is a splice gain mutation. It's synonymous, but clinical geneticists miss this all the time. But the magic is this can be treated now. And if we can force splicing away from what we think is a cryptic splice acceptor back to the normal splice site with an antisense, we should be able to treat her. So now we're really interested in seeing whether we can develop an antisense that can correct the splicing in her. And this is an example of a splice gain mutation. This is another patient. Her name is Margot. Her mom allows us to use her picture because she's on Twitter and everything. And she has an SCN8A mutation, and she has epileptic encephalopathy. She's 14 months. She presented at 3 months of life with really severe seizures, and she failed all current medications. No other organs involved. And the SCN8A mutation, this is another sodium channel, and it's exclusively in excitable cells, and this is the OMIM entry. It leads to autosomal dominant inheritance. And her mutation is in exon, what's called exon 5N. And the way SCN8A splices, it splices from exon 3 to 4. Then it incorporates either 5N or 5A, but never both. And the N is the neonatal form, and the A is the adult form. Her mutation is here in 5N, and this is the amino acid substitution. This mutation, it shows delayed activation, delayed inactivation, and persistent current. So it really is pathogenic. So normally you'd say there's no way to treat this, really. This is a bad mutation. It's some sort of gain of function mutation. But she's got this standby exon that's just sitting there waiting to be used. So the idea is, could we splice switch? So we'll create an ASO that prevents splicing to this bad exon and forces it to this exon here. And so we're excited about using that. And that's an example of a splice switching ASO. It's not I showed you before what are called poison exons, but in her case, it kind of is a poison exon. It's a bad exon. And the last example is a gene called NARS1. And NARS1 is really interesting. So NARS is the asparaginase tRNA synthetase. So it's the cytoplasmic version of the enzyme that adds asparagine to the tRNA that's required for protein synthesis in the cytoplasm. And what's interesting about this gene, if you look it up in OMIM, there's both a recessive and a dominant form of disease, which is really strange. Hardly any diseases in OMIM have both dominant and recessive, where the phenotypes are identical. You can't separate these two. So it tells us there's something special about the dominant mutations. How can there be two different zygosities for the same clinical presentation? I mean, something very strange is happening. And from this paper that was published in 2020, what was found is most of the mutations are loss of function, but there are three very special mutations indicated by this red box that are monoallelic and always de novo. And two of them are missense, and one of them is a stop codon here at the very end of the gene. So the stop codon, you might be thinking, evades nonsense mediated decay, and that's exactly what happens. And these other two are probably inhibiting the function of this enzyme, which produces a homodimer. Either prevents homodimerization, but acts as a dominant negative. So this creates an opportunity, because normally to produce disease, you have to be missing both copies. These patients have one healthy copy, but they got one bad copy that's dominant, that's a toxic gain of function. So the idea is, could we target the one bad copy with an allele-specific antisense oligonucleotide and leave the other copy intact? So that's what we're trying with this family. And these examples show you kind of the power. If we can just tune our therapeutics, if we understand something about the mechanism of the mutation, next we have to figure out when's the right time to treat. You know, what we've learned from conditions like nusinersen is that if you treat and you can measure a delta, which is the difference between the expected outcome and the improved outcome, then you can show clinical benefit. And we also learned that, you know, earlier treatment gives you more opportunity to measure a delta. There's going to be some conditions where maybe too much damage has been done, or the disease has already set in, and so it's going to be really hard to measure a delta, even if you have a therapy that is, you know, targets the mutation and is beneficial. And some of them are just too slow. We would have to wait years to see any benefit. So we're thinking a lot about this in terms of patients that are being nominated to NLORM. The FDA is really our partner in this. They have issued special guidelines for antisense therapy for ultra-rare patients, and that is a very clear path to go from a patient with an unmet medical need to an antisense that can treat their mutation within about a year. And so what NLORM is doing, it's taking nominations from tertiary care centers. They undergo review by a committee of peer academic scientists that I lead at NLORM. And if the patient is approved for therapy, then it goes into the discovery phase where an antisense is developed. NLORM then does the manufacture and sterile fill and finish and sends it to the pharmacy of the doctor that's treating the patient. It also produces the FDA document, what's called the IND, that gets submitted for the N of 1. It's ethically approved. And then eventually, 12 to 18 months later, ideally, treatment can begin, and we can hopefully measure something about outcome. And this is the ideal situation. It doesn't always work so smoothly because there's bumps, but the idea is if we can scale this up and get up to maybe 200 patients or something, we can really learn a lot. So far we've had over 300 nominations, which has been great because we never knew we were going to get nominations. And this is the current status. There's 10 that are currently under evaluation, 60 are approved where the drug is being developed, 20 are in preclinical development, so we're very close to releasing the lead ASO. About 5 of them have FDA applications that are pending, and 29 or so have already been treated. And we have a lot of support from the FDA. 4 divisions within the FDA have been supportive. And these are some of the genes that we have in our pipeline, from the ASO design and screening to tolerability in animals, GLP tox manufacturing, regulatory, and finally treatment. These are the ones that have crossed the finish line here. And what we're finding is a lot of the ASOs we make just for one patient can be repurposed and used for another patient. Just turns out they maybe have the same mutation or the same underlying mechanism. And a lot of these here, we're hoping to push them towards the finish line in the next couple of years. And this just gives you a zoom in of 2024. We started with this gene in 2024, where KIF1A, and now all these other ones, we've crossed the finish line. They're now either dosed or we're just finishing the FDA approval. So that's about 15, 20 genes in a year. This was one of the first papers that we had that was just published a couple of months ago from Wendy Chung, who was at Columbia at the time of this clinical trial. This was an N of 1 treatment for a condition called KIF1A, which is a spastic diplegia. This girl had been suffering a lot, and she was treated for 9 months here with an antisense oligonucleotide, and it was well tolerated. It was safe. Most outcome measures, including severity of the behavioral arrests, improved. And we're hoping for a lot more examples like this, because this same ASO can then be used in other patients. This was our first patient treated at Rady Children's Hospital. This child had a personalized antisense oligonucleotide for a gain-of-function mutation in SCN2A. And this is our clinician, Dr. Olivia Kim McManus, who dosed this patient, Connor. And this was featured in San Diego Union Tribune recently. So we're really excited about pushing this, and we hope UCLA is interested in this kind of thing, because it's, I think, really the future of personalized medicine. For sure, we can, develop therapies very quickly. So I won't go through the conclusions, because I've already been through all these things. And I just want to acknowledge my laboratory, former lab members, spina bifida collaborators, and clinical collaborators. Thanks so much.
Time for questions.
Yeah.
Thinking about like how long you have to
How long? Yeah. Yeah, how long do we have to treat? So most of these are structured as 1 year, N of 1 clinical trials, meaning we use the patient's own natural history to determine if there's benefit. And after a year, so N-Lorem makes the drug for the patient for free for life. And so the drug is available for as long as the patient wants it. But after a year, we consult with the doctor and with the family and see if there's been benefit. And then we hopefully will publish a paper, and report it. And the patient's then free to continue on that drug for as long as they'd like.
Question over here.
Yeah.
Hi, similar vein to that question, but when you think about treatment of these individuals, do you think about potential benefit based upon the age of the individual at, like when they're going to be able to receive the treatment, and what phenotypes can be measured, 1 year out?
Right. Yeah, I think the timing of the treatment and the natural history is very important considerations, definitely. There's patients enrolled in N-Lorem that range from newborn up to 70 years of age. That gives you some idea. And of course, we don't want to start the process if a patient will die, let's say, before the drug is ready, because that's kind of pointless. So we do take that into account. The committee that, decides which patients should be accepted into the program looks at all of those things. We did have a couple of early patients that progressed so quickly that we had to just tell the family that we couldn't develop the drug in time. But as we learn more, and the process gets faster, because now we're scaling up. N-Lorem has a whole lab. There's like 20 scientists that are working there to develop these drugs. We hope that process to develop the drug goes faster. But because they're N of 1 trials, we still have to get ideally at least a year of natural history from when the patient enrolls until the first dose. So that gives us some idea to understand the trajectory.
Great talk. I'll be talking with you later as well. But is there an optimal time, just sort of continuing the previous question? So we know that with Duchenne muscular dystrophy, there are some of the new drugs, there's a certain age cutoff where they think that you're going to get the maximal benefit. And so are there some conditions for which there is sort of that, I'm not going to say developmental window, but that ideal window where this could be maximally optimal for an influence outcome?
Yeah, how can we gear the trial so that, I think that I think the drug companies are probably better at that. You know, there you can really structure your trial to enroll patients at a particular state in their disease course. And so you can really tune it to have maximal chance. And LORM really doesn't have the benefit of that because we're just taking patient nominations. We don't tell families when in the disease course they should be nominated. And so far we've not declined any based upon the status of the current clinical status. My thinking is if we can make a drug for a patient, maybe they're too far advanced to benefit much. Maybe they'll benefit a little bit. Maybe that same drug could be used in another patient then in the future. We just don't know. It's a great question.
Let me, so wearing the sort of clinician's hat, do you think what do we need to do when, with the bioinformatics analysis of either whole exome or whole genome sequencing so that we can pick up more of these, more mutations that will affect splice variants? Because, and do we need to be doing RNA-seq as part of the standard?
I think definitely yes. We should be doing RNA-seq. But I think the other part of the solution are the clinical geneticists who read out the reports, whether it's a gene panel or an exome or a genome. Right now, all they say is pathogenic, likely pathogenic, or VUS, right? I'm really hoping we can convince them that they should score them based upon some other metric, either gain of function, loss of function, splice change, something like this to help guide the therapy. Because now it's up to us, it's up to my team to look at the report and figure out whether these could be pathogenic. There was nothing in the nomination from that splice gain that said this was a splice gain. We had to plug it in, and this is work that the clinical geneticists should be doing. They should be our partner. And I think if we had RNA-seq, that would add a lot. Yes, definitely.
Questions? Well,
Well, you've asked excellent questions. Thank you so much for coming today and for participating.